Hi, I'm Ella, and I am going to discuss about fertilization. Fertilization is a complex process and involves a cascade of events. So here are the sequence of events leading to fertilization. First is the hyperactive motility, leading to the binding to the zona pellucida, acrosomal reaction, penetration of zona pellucida, sperm-oocyte membrane fusion, the sperm being engulfed, the condensation of sperm nucleus, and formation of male pronucleus. Firstly, as capacitation is completed, the motility patterns of spermatozoa in the oviduct become hyperactive. The sperms swim like an Olympic swimmer, or they swim in a frenzied or dancing manner. Next is the binding to the zona pellucida. The first binding site, referred to as the primary zona binding region, is responsible for adherence of spermatozoa to the zona pellucida. The second binding site on the spermatozoal plasma membrane is believed to be acrosome reaction promoting ligand. This is much like a typical hormone receptor binding complex. The binding initiates the acrosomal reaction. Next is the acrosomal reaction. This reaction begins when the plasma membrane of the spermatozoon forms multiple fusion sites with the outer acrosomal membrane. When the two membranes fuse, many small vesicles are formed. This process is called vesiculation. After vesiculation has occurred, the acrosomal contents are dispersed and the sperm nucleus is left with the inner acrosomal membrane surrounding it. So there are two purposes of the acrosomal reaction. First, this enables the spermatozoa to penetrate the zona pellucida. Second, it modifies the equatorial segment so that it can later fuse with the plasma membrane of the oocyte. Next is the release of acrosomal enzymes. Following attachment to the zona pellucida, the acrosome reaction allows the release of a variety of enzymes. Acrosin, released from spermatozoa during the acrosomal reaction, hydrolyzes zona proteins as well as enhances the sperm's ability to bind the zona. The acrosomal reaction allows the spermatozoon to digest a small hole through the zona through which it can pass. Fifth sequence is the fusion of the equatorial segment and the oocyte plasma membrane. When the spermatozoon completely penetrates the zona and reaches the perivitelline space, it settles into a bed of microvilli formed from the oocyte plasma membrane. The plasma membrane of the oocyte fuses with the membrane of the equatorial segment and the fertilizing spermatozoon is engulfed. Next is the cortical reaction. After membrane fusion, the oocyte undergoes a series of changes that prepare it for early embryogenesis. The most easily recognizable is the cortical reaction. Exocytosis of the cortical granules result in the zona block. Zona block is a process where the zona pellucida undergoes biochemical changes so that further sperm cannot penetrate it. Polyspermy is prevented by the zona block. Finally is the pronuclei formation. After the sperm nucleus has entered the cytoplasm of the egg, it becomes the male pronucleus.
So now I am going to talk about the delivery of semen to the proper anatomical region of the female tract that is required for a successful artificial insemination. So there are three techniques for a successful artificial insemination. First technique is the TRANSCERVICAL insemination. So it is a artificial insemination technique that requires the spermatozoa to be deposited in the reproductive tract of the female by artificial means. So in general, semen is delivered using a pipette to penetrate and bypass the cervix. Then the next technique is the intracervical insemination. So in the sow, the insemination pipette is positioned within the cervix and semen that is delivered into the cranial half of the cervix and flows directly into the uterine horns. And lastly, the intravaginal insemination. So in dogs and cats, semen is deposited in the cranial vagina.